What? Last episode already? Let's get to it. So, for the coronavirus, we've understood the basics, the virology, the history, the immunology, and now we are ready to learn the specifics, the origins, and further scientifics about the coronavirus. Now, we aren't focusing on the political aspect of the coronavirus, as many have chosen to do. Instead, we are going to learn the facts that have created such a controversy. But mostly, it's just the exciting and interesting stuff we'll be learning today. Before we begin, let's mention the name of the coronavirus. The scientific name of the coronavirus is SARS-CoV-2, or in other words, it's Severe Acute Respiratory Syndrome Coronavirus 2. This became the name of the coronavirus because there was a genetically similar coronavirus similar on in the 2000s called SARS. And this is basically the second version of it. The coronavirus is also called the Wuhan Coronavirus because it is said to have started in Wuhan, China. And to put it more specifically, SARS and the coronavirus are very similar. The coronavirus began at the end of 2019 around December. The first case was documented on December 1st. So the 19 in COVID-19 meant it started in 2019. But anyways, if you've done a simple search of where the coronavirus began, you might have come across cases originating from the Huanan seafood market. In fact, there was continuous reports that discussed pneumonia cases back in December 2019, and in the end, the seafood market closed the first day of 2020 in order to clean the market. This market was suspected to be the origin of the coronavirus, and eventually, these suspicions led to a simple conclusion. However, this was not the case, and I don't mean the corona case. Reasons behind the controversy of whether or not the virus originated from the market was based on the variety of observations from the market and to what we know of the coronavirus of right now. To be more specific, the first patient to show these coronavirus syndromes was on December 1st, and this person had no contact or relation with the market at all. The second reason was how there was no epidemiological link or skin-to-skin contact with this case to others, which meant that if the conclusion about the seafood market was true, this would mean that the first case would have had to either gone to the market or would have had physical contact with anyone from the seafood market in order to get the virus. The third reason was the fact that there were no bats found or sold within the seafood market. And why are we talking about bats? Well, after analyzing the genome sequence of the coronavirus, a research paper was submitted about how there was 2 bat viruses that were closely related to the coronavirus. The 2 other viruses were sampled from bats and were the co-VZC45 and the co-VZXC21. In fact, the paper stated that there was an 89.1% similarity in nucleotides with the co-VZC45 virus, and it also had 100% similarity in the amino acids in the proteins used in the coronavirus. This was checked and supported by the use of BLAST, a basic local alignment search tool. What BLAST does is it helps align and compare certain protein or nucleotide sequences, and it shows how similar one thing is to another. Actually, I did a lab with this website, and I learned that even though the percentage of 85 or 90 shows that the genome sequence is extremely close between 2 things, I found that one little difference in percentage could differentiate 2 whole species. To be more specific, by aligning the genome sequences of humans and chimpanzees, we have a 96% similarity. Just remember, the differences in percentages may be a little number, but in reality, the differences in phenotypes, or how you look, appear, and act, are numerous. Now, even though there is only an 89.1% similarity in nucleotides with the co-VZC45 virus, the 100% means the amino acids in the percentage are exactly the same. So, the coronavirus also has an 88% similarity to the SARS-like coronaviruses that came from bats in other parts of China. Even one of these SARS-like coronaviruses has 100% amino acid similarity in regards to the proteins and amino acids. It's a little specific, I know, but if you're interested, I am. From this research, it can be shown that SARS and bats are both linked and related to the coronavirus. This further shows that the virus has jumped species, and the 100% in amino acid similarity hints that it may have been a man-made virus. Though, before I go on, I just wanted to let you know that this is the controversy and a theory many have. I have a neutral stance on the controversy, and I don't intend to offend anyone. Okay, now back to learning. So let me tell you a little about the SARS virus back in 2003. SARS stands for Severe Acute Respiratory Syndrome Coronavirus, and it started in the Huanan province of China. It had spread to about 26 to 27 countries within 5 months and killed about 800 people. The first case was on November 26, 2002, and research found that this case and other early cases had a history with wildlife. And when the researchers and scientists analyzed the civets in the market, they detected that the civets had the same SARS virus as humans. And furthermore, scientists believe that the SARS virus in civets came from a bat's reservoir. A little extra information, when I say a bat's reservoir, I'm describing how a virus like SARS is able to have a commensal relationship with its host for a long period of time. So the SARS virus stays alive while the virus and the host are at peace with each other. And the virus evolves over time as well. And so the bat is basically a reservoir for this virus. Okay, so the civets weren't the source of the virus. Instead, they got the virus directly from the source, and reasoning behind it shows how the farms with civets across the country had the SARS virus too. So it is definite that the bats are the natural reservoirs of the SARS-like coronaviruses, but we are not exactly sure if it is the natural reservoir of SARS itself. Overall, this also shows the virus going across species. The coronavirus has come from fruit bats to humans.
There is no definite information of any animals in between to have gotten the virus, though there is a definite cross-species transmission, also known as a host jump, spillover, or interspecies transmission that has occurred. And it's not easy to do this because viruses that go through cross-species transmission have to cross the species barrier. In order to do this, there must be specific mutations to the receptor specificity that allow for the virus to change hosts. Though, through my research on RNA viruses and other RNA viruses like the flu, this could occur by humans repeatedly interacting with infected animals or the quick reassortment of genetic segments from virus strains to make a new virus. Though, through the mutating process, the virus's receptor is very important. The coronavirus's S strain has high homology with the SARS virus. From past videos, I have explained what the S protein is, but I will give you a small reminder right now. The S protein or
the spike protein are attached to the virus's surface, and they are basically the key in the lock and key theory, where the virus uses a key to invade cells. Without it, the viruses wouldn't be able to enter human cells and infect us. In fact, as I've said before, the viruses evolved like humans. They adapt and mutate in order to stay alive. So, the viruses that have evolved over time, they are able to infect humans, while the viruses that haven't evolved over time aren't able to infect humans. And a scientist who's been researching a virus's ability to infect other species is Dr. Shi Zhanli. Dr. Shi Zhanli is a Chinese virologist and was the one who discovered the spike protein's role in the transmission between species. In a paper she wrote in 2010, she discussed the angiotensin converting enzyme 2 proteins, or ACE2 proteins, and the relationship of it to the S protein in SARS. ACE2 is an enzyme found specifically on cells living on air sacs within the lungs that is attached to the cell membrane and it lowers blood pressure. Dr. Shi Zhanli found that there is a great diversity of ACE2 within bats, more than other species, which makes many more bats to act as a reservoir of SARS. Based on other articles that I've researched, the ACE2 receptor acts as the door and the lock for the spike proteins to open. The spike protein picks the lock of the ACE2 receptor, and this is how the spike protein is able to enter the cell. The spike protein has 3 components that make it look like a rocket. So, in the process of invading the human's lung cells, the spike protein would attach to the ACE2 receptor before the boosters of the rocket come off. The space shuttle of the spike protein would change its shape to act as a lock in order to merge with the membrane of the cell and therefore have the ability to inject the virus's genetic material into the cell. If you want to know even more specifics, there's another interaction, as we know of right now, or a month ago, involved with the infecting of the coronavirus into the lung cells. This is the TMPRSS2, or the transmembrane serine protease 2, which is able to compare the spike protein shape so the spike protein can unlock the lock. The TMPRSS2 is found on the host cell and is needed for the coronavirus to enter the cells. It is supposed to act as the security or the inhibitor for the cell before any lock could be unlocked. But in this case, it activates the spike protein so the spike protein could successfully attach itself to the host cell. Anyways, research from Dr. Shizhan Li also helped with more information regarding the cross-species transmission. In fact, she found from SARS that there doesn't need to be a middle host, and the transmission could simply be from bat to human. So, there isn't a bridge of animals the virus has to cross in order to get to the humans. When the Wuhan outbreak occurred, researchers from all around the world were trying to uncover the mysteries of the coronavirus. When Indian researchers began to analyze the genome sequence of the coronavirus, they found a similarity in the spike protein in the coronavirus and the HIV GAG protein, which is the HIV1 GP120 and GAG, which helps in the process of multiplying the virus and putting the virus together. In other words, it basically helps during the assembly, and then once the virus is injected into the cell. They also found, through GenVake, that the coronavirus had 4 new sequences inserted into its genome, and all of these new sequences were from HIV. This is when the controversy once again erupts, as scientists and researchers find it quite mysterious and shocking as to HIV similarity in the coronavirus sequence. Overall, coronavirus, HIV, and that coronavirus, which was discovered by Dr. Shizhan Li, have all 4 of these sequences. Another researcher has found that there are structural similarities between HIV's GP41 and the coronavirus's spike proteins. This makes sense, as the GP41 is a protein that acts as a key that results in the failure for the immune system to work, and it's very important in regards to infecting cells. Overall, the world is still researching the mysteries behind the coronavirus. At the same time, they are trying to find cures and vaccines. As I said in the last video, science and the medical world, they're still developing, and time and patience is needed for a vaccine to come out. Some scientists and companies have tried to take pieces of the genetic code for the S protein and inject it into the body with fatty non-particles. Others have looked into the past history for a vaccine. There have also been trials and controversies with hydroxychloroquine, an immunosuppressive drug, and remdesivir, an antiviral medication that stops the spread of severe coronavirus infections, over whether they in fact cure and destroy the coronavirus altogether in the body. Though there isn't an exact cure or antiviral treatment as mentioned by the CDC, and all we need is time
and patience to find a cure. On the positive side, there are some ways to know if you do have the coronavirus so you don't spread it to others. Once the symptoms start showing after 7 to 14 days of being introduced to the virus, you could self-quarantine and isolate yourself from others. And if you're unsure, there are tests that you could take in order to show if you have or had the coronavirus.
The first of 2 are molecular tests, which find any active infections occurring regarding the coronavirus. The first test is the Saliva Test, where you spit into a tube to a certain line before you put the cap on the tube and mix some blue solution with the spit in the tube. You can't eat or drink anything, or even brush your teeth, for 30 minutes for the saliva test, as the substances in your mouth can mess up the results of the test. Though, the test is quick and easy, and you'll find your results within a few days. The next test is the nasal swab test, which is super uncomfortable. I know from experience, trust me. What occurs is the doctor or nurse sticks a 6-inch pencil-like swab up your nose until it reaches the back of your throat for 15 seconds before it gets taken out. Once both nostrils have been infiltrated with the long swab, the nurses and doctors will send samples in a container to the lab, where they'll notify you in as little as an hour for the results. The third test is if you had the coronavirus at one point and the scientists and doctors are merely checking for the antibodies of the coronavirus. It is a serology test, and it's a blood sample either by a finger prick or by drawing blood from a vein within the arm. The finger prick is less accurate, however, so it is preferred to draw blood from the vein. The sample is then brought to a lab to be tested, and it could take from a day to weeks for results to come out. It just really depends on where you've taken the test. Overall, the coronavirus is the pandemic of 2020 and a little bit of 2019, and we must remember all that occurred so we can avoid such a pandemic again. So many lives were lost, but not as many as the past pandemics. We are learning to stop the spread of the virus, along with how to protect and prepare for it. I hope from these videos you've truly learned about the coronavirus from the inside out, and I hope you understand why the world did what it did, and what the coronavirus really is. A germ. A virus. And it's as simple as that. Thank you, and have a nice day.